आज आम डिस्कसन करवा प्लांट टिश्यू कल्चर विषय। तो ये वीडियो टे देखापी तो यहीपरी आउ अन्य वीडियो मान को भी देखापी वीडियो को लाइक करूँ, शेयर करूँ, सब्सक्राइब करूँ। तो जहाँफल कि मुझे जिते वीडियो ये रिलेटेड छाड़ी तो सब देखिपारे। ओके, तो ये आम डिस्कसन करवा व्हाट इज टिश्यू कल्चर, व्हाट आर द बेसिक रिक्वायरमेंट ऑफ टिश्यू कल्चर, हाउ टू मेंटेन कल्चर रूम एंड other माने cultures in tissue culture. Okay, what is tissue culture? Tissue culture is a technique which is used to produce a large number of a plant by using small part of plant or called explant in laboratory condition or sterile condition on a nutrient medium. And the plant are produced in tissue culture is called tissue culture raised plant or micropropagated plant or in vitro regenerated plant. Different terms are used in tissue culture are given like acclimatization basal medium, Carlos, clone, explant, in vitro, inoculation, micropropagation, organogenesis, advantageous organogenesis, rooting, in vitro regenerative plant, then protoplast regeneration, suctive culture, somaclonal variation, somatic embryogenesis, stock plant, subculture, suspension culture, totipotency, transplant stage, synthetic seed technology. Then what are the equipments are required in a plant tissue culture like electronic balance, pH meter, autoclave, laminar air flow. Then these accessories are required inside the laminar air flow. Then micro oven, hot air oven, distillation unit, fridge. These are glassware required for plant tissue culture like culture bottle, conical flask, beaker, measuring cylinder, pipette, culture tube. Then plastic wares are required for plant tissue culture like distilled water jar, wash bottle, multiple potting tray and tray. Then other accessories like cotton plug, absorbent cotton, non-absorbent cotton, tissue paper, brush, gauze. Then culture room condition, how to maintain culture room condition. Culture room condition should be maintained in aseptically or sterile condition by always closing the door, and the culture room condition temperature is maintained 24 or 25 degrees Celsius plus minus 1 degree Celsius. The light duration should be maintained 16 hour light and 8 hour dark, and the light intensity should be 3000 lux intensity. The aseptic condition hot and chilly glassware nutrient medium culture room inoculation chamber must be maintained in aseptic condition. Generally microbes are compete with growing tissue and finally kill the tissues or explants. Therefore we should maintain aseptic condition in and around the culture equipments in tissue culture experiment. There are three sources of contamination. What are these? Now these are through potential or culture medium. First thing. Second thing, through explant material. And the third is through inoculation. During we inoculate the explant in the culture medium, it should be contaminated. Then how to avoid the contamination through utensil? By surface sterilizing with sodium hypochlorite or calcium hypochlorite or chlorine water or mercury chloride, or by autoclaving at 21 degrees Celsius for 30 minutes for complete sterilization. But the time duration of autoclave varies according to the size of glasswares and the volume of solutions or chemicals or medium. Then microorganisms get killed at this temperature by autoclaving, and explant how to sterilize the explant. Explant can sterilized or washed by thoroughly washing under running tap water. Then these are treated with liquid detergent like Twin 20 or Tipol. Then followed by it can be sterilized or washed by using fungicide like Babistin. Then rinsed with distilled water for 3 to 5 times. Finally, it is treated with 0.1% mercury chloride inside the aseptic chamber, and then it is treated with or rinsed with autoclaved distilled water. To prevent the entry of microbe at the time of inoculation, laminar airflow cabinet or chamber used for culture is sterilized with UV light or filter sterilized air. The principal advantage of working in this cabinet is that the flow of air does not hamper the use of spirit lamp under tropical and subtropical areas where dust particles are very high. It must be kept in a double door fitted culture room in order to prolong the effective life of the filter. The culture vial containing medium are plugged with non-absorbent cotton wrapped with cheesecloth or gauze. Such a closure allows the exchange of gases but does not permit the entry of microorganisms into the culture vials. Then nutrient medium. What are nutrient medium? The mediums are required for growth of explants into a plant is called a nutrient medium. The composition of nutrient medium is varies or a different type of nutrient medium are used in a plant tissue culture like MS medium means molasses and skog medium, B5 medium, N6 medium, woody plant medium, etc. The supply of energy and essential metabolites to grow the tissue within the state of culture medium, solid or callus culture or liquid or suspension culture. Solid culture means the medium with gelling agents occur, and the liquid medium means the medium without gelling agent. What are the ingredients of a nutrient medium? The ingredients are major and minor elements or micronutrient and macronutrient, carbon source like glucose, sucrose, table sugar, sorbitol, maltose, etc. Then vitamins like thiamine, pyridoxine, niacin, etc. Then hormone or growth regulator are used for shoot regeneration or rooting or callus culture like oxygen, cytokinin, gibberellic acid, etc. Out of all these medium, MS basal medium is called the choice of a medium in plant tissue culture. Why? This medium is frequently used for plant tissue culture. These are the composition of two growth media, but here given some micronutrient and macronutrient and vitamins for MS basal medium. Then steps of tissue culture. In tissue culture, different steps we followed, like a selection of mother plant means a healthy, disease-free, elite plant. First, we select before tissue culture. Then we maintain it in the garden or in their natural habitat or in the garden. Then collect the explant and it is inoculated, surface sterilization and inoculated for shoot initiation. Then it is subculture for multiplication of a shoot, and then after shoot multiplication these shoots are taken and inoculated on a rooting medium for root formation. After rooting, in vitro regenerated plantlets were acclimatized. in the soil, either in the net house or greenhouse. Subsequently, these are transferred to the field. Then type of a tissue culture. Different type of a tissue culture means the pathway of tissue culture, like a meristem culture, direct adventitious organogenesis, callus culture, somatic embryogenesis, synthetic seed technology, then hairy root culture, then cell suspension culture, different type of culture, embryo culture, etc.